Welcome to the fascinating world of parasite ecology. Imagine you are an astronaut landing on a completely alien planet. To survive, you need to understand the temperature, the atmosphere, and the local creatures that might try to attack you. For a parasite, that alien planet is the body of its host. Today, we are going to explore how these tiny invaders survive, thrive, and interact with their living, breathing environments. It is a fascinating double life where the environment is actually fighting back. Now that we have introduced the idea of a host as an alien planet, let us look at what parasite ecology formally means. In scientific terms, it is the comprehensive study of the interactions between a parasite and its multiple environments. Unlike a free-living animal, like a deer, that only worries about the forest, a parasite actually exists within two distinct ecological realms simultaneously. They have to worry about the host's immune system, the nutrients in the gut, and how to get their offspring out into the wider world. Understanding this ecology means studying how parasites navigate both of these environments to find mates and infect new hosts. Building on this idea of two simultaneous realms, let us define them clearly. The first realm is the microenvironment. This is the specific living habitat inside or on the host. It could be the highly acidic stomach, the oxygen-rich bloodstream, or the surface of the skin. The second realm is the macroenvironment. This is the external physical world where the host itself lives, such as a dense tropical rainforest or a calm freshwater lake. A successful parasite must be adapted to survive the hostile, living microenvironment of its host, while also being able to navigate the broader macro environment outside to spread. To make these two realms perfectly clear, let us look at this diagram. Here we see a fish swimming in a lake. The lake itself, including the water, the soil at the bottom, and the aquatic plants, makes up the macro environment. This is the external world. But look inside the fish. We see a cutaway showing the gut and the bloodstream. This is the microenvironment, the host body where the adult parasites live and feed. Notice the arrows showing the life cycle. Infective stages enter from the macroenvironment into the microenvironment, and later, parasite eggs are released back out into the macroenvironment. This diagram beautifully captures the dual habitats a parasite must conquer. Having seen how parasites move between these environments, we must ask, why does studying this matter in the real world? The answer is simple. If we do not understand where a parasite lives and how it interacts with its environment, we are completely blind when trying to stop it. For example, by understanding the macro environment of the malaria parasite, we know it relies on mosquitoes breeding in stagnant water. This allows us to control the disease by draining swamps or using mosquito nets, rather than just relying on drugs. Furthermore, as climate change alters global temperatures, this ecological knowledge helps us predict which new regions might become vulnerable to tropical diseases. Now that we understand the environments parasites live in, we need a way to measure their success. To truly understand the ecology of a parasite, we cannot just use words. We need to measure how successful the parasite is at infecting a host population. Ecologists use specific mathematical formulas to track these population dynamics. The two most fundamental measurements we use are prevalence and mean intensity. Prevalence tells us how widespread the infection is across the entire population. On the other hand, mean intensity tells us how severe the infection is specifically within the hosts that are actually sick. Let us dive into the mathematics of these metrics. Let us look at the formula for prevalence first. Conceptually, this formula simply calculates the fraction of the population that carries the parasite. The formula is P equals I divided by N multiplied by 100. Here, P represents the prevalence, which is usually expressed as a percentage. The variable I stands for the number of infected hosts in the population. The variable N represents the total number of hosts examined, including both healthy and infected individuals. If the number of infected hosts increases while the total population stays the same, the prevalence will increase. This physically indicates that the parasite is spreading more widely through the macro environment. Before we move on to the next metric, let us see this with a concrete example. Suppose you are a wildlife biologist studying a lake ecosystem. You catch and examine a total of 50 fish to check for a specific parasitic worm. Out of these 50 fish, you find that 10 are infected. We want to find the prevalence. We are given N equals 50 and I equals 10. We choose the prevalence formula because we want to know what percentage of the fish population is acting as a host environment. We substitute 10 for I and 50 for N. Dividing 10 by 50 gives us 0.20. Multiplying by 100 converts this decimal into a percentage. The final answer is 20%. Physically, this means 1/5 of the fish in this lake are infected with the parasite. Now that we have calculated the prevalence, we need to know how heavy the burden is on the infected individuals. This brings us to mean intensity. A host might have just one parasite, or it might have 1,000. The formula is M equals T divided by I. The symbol M represents the mean intensity, which is the average number of parasites found per infected host. The variable T is the total number of individual parasites counted across all the infected hosts. The variable I is again the number of infected hosts, completely ignoring the uninfected ones. If the total number of parasites increases, but the number of infected hosts remains constant, the mean intensity goes up, meaning the infection is becoming much more severe and crowded for those who are sick. Let us return to our fish example to calculate this new metric. When you carefully count the worms inside those 10 infected fish, you find a total of 120 worms. We are given T equals 120 and I equals 10. We use the mean intensity formula to figure out how crowded the worms are inside the infected fish. We substitute 120 for T and 10 for I. We are dividing the total worm burden evenly among the fish that actually have worms. 120 divided by 10 gives us 12. Physically, this means that if you catch an infected fish in this lake, you can expect it to have an average of 12 worms living inside its gut. Having measured the current state of an infection, we must now look to the future. In parasite ecology, it is crucial to know if a parasite population will survive, grow, or die out when introduced to a completely new environment. This potential for growth is measured by a critical metric called the basic reproduction number. This number helps ecologists predict the future of an outbreak. It takes into account how fast the parasite transmits, how crowded the host population is, and how long the infection lasts. Understanding this concept is vital for predicting whether a newly introduced parasite will simply fade away or cause a massive ecological disruption. Let us look at the mathematical form of this concept. The basic reproduction number for a macroparasite in a simple ecological model can be estimated by multiplying 3 key factors. The formula is R0 equals beta times C times D. Here, R0 is the basic reproduction number. The symbol beta represents the transmission rate, meaning new infections per day. The variable C is the host density, or how closely packed the hosts are. Finally, D is the duration of the infection in days. If any of these variables increase, R0 goes up. Crucially, if R0 is greater than 1, the parasite population will grow and establish itself. If R0 is less than 1, the parasite will eventually die out because it cannot replace itself fast enough. Let us apply this formula to a real-world scenario. Imagine a new species of tick is introduced to a deer population. We are given a transmission rate, beta, of 2 new infections per day. The host density, C, is 0.5 deer per square kilometer. The duration, D, is 4 days. We want to calculate R0 to determine if the tick will establish itself. We substitute our values into the formula. First, we multiply the transmission rate of 2 by the host density of 0.5, which gives us 1. This represents the effective contact rate per day. Finally, we multiply by the 4 days the tick stays on the host. The final answer is 4. Because 4 is much greater than 1, the ecological interpretation is that every single tick will give rise to 4 new infections, and the population will rapidly explode. We have covered a lot of ground today, so let us review the key takeaways. First, we learned that parasites live a unique double life, constantly navigating the hostile microenvironment of their host and the broader macroenvironment outside. Second, we saw how ecologists track these invaders using math. Prevalence tells us how widespread an infection is, while mean intensity reveals how severe the crowding is inside the sick hosts. Finally, we used the basic reproduction number to predict whether a new parasite population will explode or die out. Understanding these ecological dynamics is the absolute foundation for controlling parasitic diseases and protecting global public health. You now have a solid grasp of how parasites interact with their complex worlds.